Okay. Yes, yes. Hello. Uh, I wish you are good, and we want to start our class. For the last session, most of you say to me that your book is very hard, but I think it was good because all of things you need to study is there. Okay, and I take exam only from that, but not all of them. I can show you PowerPoint or a slide, but you can note yourself. And it was, I think, difficulty than that way. Now, do you understand what I said? I want to explain about your book. Muhammad, what do you like? Okay, only. Okay, you don't need to study all of them. Only study the thing that I say you in end section. Okay? I'm waiting for your slide. Please wait. Okay. What do you mean material? What do you mean material? You mean gram strain? Do you mean gram strain, Marbe? Please say, tell me. Do you mean gram strain? Salam Asra. What do you mean about material? Please say me. Material, what do you mean? What do you mean about Marve? Do you hear me? What do you mean about okay, okay. I tell you about this, but I want only learning you the here, please look. Pelagia. Okay, here look, for example. Please look. Okay. I want to speak this section, this term about this flagella, pili, capsule, plasma membrane, cytoplasm, cell wall, liposaccharide, teichoic acid, inclusion, and spores. Okay, I want to speak this, teaching you this. Okay, but in here there are in Persian. I give you one book or lecture that have in English and you can only study that. You didn't need any more writing or read. Do you understand what they mean? Come on here, for example. Just a moment, wait. It is loading. Okay, here, please look. About, for example, about cell structure. I can do someone for you, okay, for help you. I can't highlight the where you should read or study for exam, okay. In section, I can highlight, for example, in this you didn't need to study this paragraph. Only study this. I can highlight for you in next section. I don't know what should I do, Marve. Please go out and come back. Okay? Just a moment, please listen to me. This slide, this book is very, very complete for you and is study very easy. Okay? I can't highlight for you in last section where you should study. Not all of this. The where you should study, I can give you and want you to study. That's okay? Are you okay with this? Okay. I can help you like this. Highlight the book and say you only study this. Now you should only listening. Yes? Are you okay? We start? Okay, okay. I will help you like this. ببین برق نیومده مطمئنی؟ چون من با آسانسور اومدم بالا Okay, you hear me? I come back. My system is discount. Yes, I will refer you. Whatever, hey, that don't worry. Only study, only listen to me. Don't write, don't anything. Only listen to me. I didn't teaching you all of this. I never. Okay, I never. Okay, study me and listen to me. And if you have question from me, please say I answer you. Membrane. You know, all of cell have membrane outside them. The cytoplasm membrane of both gram-negative and gram-positive cell is a lipid bilayer. It is bilayer. Composed of phospholipid, glycolipid, and a variety of protein, such as protein. The protein in cytoplasm membrane, what they do, they transfer the metabolites to cell or ion cell and out. Okay? Cytoplasm membrane may extend through its entire thickness. Some of these proteins provide a structural support to the membrane and will other function in the transport of sugar, amino acid, and other metabolisms. This is why I say to you right now. What I say to you right now. The outer membrane of negative cell contains a relative high content of lipopolysaccharide. Please attention here. In gram-negative, you have lipopolysaccharide. If you want write. In gram-negative, you have lipopolysaccharide in membrane. What is lipopolysaccharide? This is a lipid containing or compound response. One of the most important identifying feature of gram-negative cell. Why? It is very important in negative cell. Why? Because of O antigen. O antigen is a epidemiology. For example, it is a important point of immunologic site in bacteria. Do you understand what I say? They are form of external polysaccharide chain and lipopolysaccharide. This lipopolysaccharide containing compounds also display endotoxin activity. Okay. O antigen is on immunologic factors, okay, or endotoxin, it is endotoxin activity, and it is responsible, it is shock observable to severe infection, and it can be case of bacteriophage in cell. For example, virus can attach to bacteria because of lipopolysaccharide in gram negative. Do you understand? If you have question, please ask me or I continue. Okay. I told you last section about this. Here, this is Streptococcus pneumoniae. And what is this around of this? I told you. What is this? What is this? This white. What is this? I told you. Capsule. Yes, Roya. Thanks a lot. Thanks a lot. Do you know how bacteria or microbe moving? How bacteria can move? Do you know with which can move? Do you know? Please say to me how bacteria can move. Okay. Flagell have some section for help this. One of this is basal body. Basal body, you know. Basal body, you know here. Many microorganisms, it say here, please pay attention here. Many microorganisms are moving, that is able to move from place to place in concert manner. It means they can move easily because they have flagella. Flagella is a simple strand of flagellin. You know, it's a simple strand of flagellin. Here, please look. Move into helical. Okay, and flagell, which structure? Okay. It's from basal body and it's from basal body. Basal body is a motor, like motor engine, okay, from flagellum. And hook, it has hook, and filament. Filament is like hair. Okay, one hair. Okay, let's listen me. Flagell include three structure. One is basal body. Basal body is motor. Is motor. Another is phalangium. Filament. It's filament like hair. And another is hook. Hook is one interloper to basal body to flagellum. Do you understand what I say? Okay, do you have any question for me? Do you have? Hook. Hook is like, I don't know in Arabic what they say. Please, just a moment, wait. Can I translate for you or no? Just a moment, wait. Hook means this. Khotaf. You say khotaf? Yes, say that. Khotaf. Hook means khotaf. Okay. Okay, so you understand what is flagell. Flagell is necessary for moving of bacteria. It is from three structures: basal body, hook, and filament. Okay? Okay? That's okay? That's right? I really explain more for you because I don't want you don't understand. If it's not enough, please tell me. I explain more. Okay. Pili. What is pili or fimberia? In many bacteria, you have external structure. They are shorter and rigid than flagella. They are very small. You know, peel around of bacteria is like a minion. You know minion cartoon? Do you see? Do you watch minion cartoons? Please, just a moment, think about their hair. Their hair is like pili. If there was bacteria, Boston, for example, their hair is pili. Do you understand my exam? Do you understand what I say? No, Mustafa, don't tell me, okay. I think I've made for you one example. I told you Pili is like hair of minion. Okay. They are very, very shorter and more rigid than flagella. This structure have been termed pili. Pili. Here, look, their hair. They are very, very short. They are necessary for sexual in bacteria. Okay. Pili play a role in cellular adhesion to surface or to host cell. Please here. Please here. Look here. Generalized or common pili. We have two kind of pili. One of them is generals. One of them is common. Okay. Pili play a role in cellular adhesion. For example, for sex. From where? From surface of bacteria to host cell. Okay. Okay. Do you understand? Do you have any question? Okay. What is ribosome? You know what is ribosome? Ribosome, what to do? What do ribosome in cell? You know? What is ribosome and what do? Please? Do you know what is ribosome and what do? If you don't know, please write no. Ribosome is very, 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 very important for what? For transcription. They are the genetic material, yes, Fatima. There are a granular appearance observed in many electron microorganisms. They are very teeny particles called ribosome. Ribosome contain 6,500 RNA and 3,500 protein. Yes, Marde, they are necessary for protein synthesis. Okay. Look here. I think we have their okay. Here, look here. This is flagella for moving of cell. Flagell for moving of cell. And we haven't here pili. Flagell is 1 or 2 or 3 in membrane. But pili are very, very a lot. Around of cell. Okay. Here, look here. It's very nice picture. This is what? What is this? Please say to me, what is this? A. What is picture A? Yes, flagell. And what is this? What are these? Yes, they are pili. Okay, okay. Look here. Flagell is one or two or maybe 10. Okay, but pili is 100 or 1,000 here. Number, I mean number. Flagell is very long, very tall, but pili is very short. You know? Okay. Do you have any question? The ribosome orchestrates the polymerization of amino acid into protein. It means ribosome make protein strain. Okay. From what ribosome make? From what? You know? Yes, that depends. From what? From what? From what? From mRNA. Ribosome make protein from mRNA. Okay? You understand? Do you have any question? Okay. They more or less several ribosome articles. They are important. They are not important. Ribosome have two subunits. 70S means a subunit. Okay. Let's go next. Here. Here. Please see here. In prokaryote, we have ribosome with two subunits. What calls 50 subunit, 50S, and one calls 30S. But in eukaryote, you have ribosome from two subunits, 40S and 60S. You understand? It is different from between ribosome of prokaryote and eukaryote. In prokaryote, we have 70S ribosome, which has two subunits, 50S and 30S. But in eukaryote, we call ribosome 80S. It's from 40S and 68 subunit. Okay. Just only for 5 minutes rest. I'm here. If you have question, please ask. Here, do you hear me? Okay. I only explain you this section from picture. It is important for you here. You know how what do you mean numbers? Okay. No. It's not important about eukaryote is 80 and prokaryote is 70. Yes, Fatima, it's right. Okay, no. We don't add them. Okay, we don't add them. This is only one number. Only one name. No, this is not important. Don't collect. Okay. How we make protein from DNA? At first, we should replication here, please see here. We have three stages from DNA to protein. One is replication, two is transcription, and three is translation. It means double-strand DNA should replicate from self and other DNA. We call them daughter cell and two daughter cells. And then from DNA, we make what we make mRNA with replication. And then with transcription, we make translation. We make protein from mRNA. I don't complete more here because it is important for your lecture. I only want to tell to you, we make from DNA to another DNA Please write this. Okay, please write. Second, you make from DNA mRNA with transcription. Okay, look here. Look here in a slide. Please look here. Look here. Yes, look here. This is replication. You have two strand DNA that are separate, and other two DNA strand make from this. Okay, you understand? They are daughter strand. This and this. Okay, you understand? Yes, there are. Yes, you're right. Okay. Next, this is transcription. Look here, transcription. This is mRNA. This is ribosome, excuse me. This is ribosome, two subunits, and this is mRNA. mRNA makes from DNA. This is DNA. DNA here in transcription make mRNA. And mRNA in ribosome with tRNA, they are tRNA. Translate protein, amino acid. Okay, do you understand? Replication, transcription, and translation. Do you understand? Okay, I will repeat for you. Do you know what is replication and transcription and translation? Do you know what I mean? What is this? Okay. This is replication. From two strand DNA, we make another two DNA with replication. Okay. Okay, Fatima. And we have transcription and translation. What is this? Transcription means make mRNA from DNA. It means like this. This is DNA. Two-strand DNA. Look, with transcription and ribosome and RNA polymerase, we make one strand mRNA. This is mRNA. You understand, Fatima? Okay. And what is this? This is translation. It means from mRNA, one strand mRNA, we make protein and we help tRNA. tRNA help us to this. Do you understand? Fatima, do you understand? Okay. Okay. Let's go. It isn't important. Don't read. Okay. This isn't important. Don't read. Don't read. I told you all of them you don't need to study. I don't want these. They're important. They aren't important. They aren't important. Please wait. Okay. And what is this? Okay. It is important. Okay. Page 90. Endospore. Do you know what is endospore? Yes. Do you know what is endospore? If you don't know, please write no. Endospore. Okay. Here. A few bacteria such bacillus and clostridium, which they are not aerobic, have a special structure called endospore. What is endospore? Endospore bodies that stain with ordinary dyes or appear to unstained highly refractory area. They are very hard to stain. Okay, they are I told you they are soluble, but they are very hard, and they are necessary for bacteria in hard position like hot, light, cold, light, dry, bacteria, not or microorganisms not die. It makes itself endoscore, means it makes outside of themselves outer coat observed as a much darker area or a thick inner coat. Do you understand what I say? No? All of you know. Okay, I'm gonna only say yes, 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 yes. Here, look here in picture. I told you it's, I think it's better. Okay, here. This is endoscope. You know what is this? This is endoscope. What? And there are 1, 2, 3, 4, 5 layers out of this. This is thing, no? We call this coat. Please write this, girls. Please write this. I will explain you. Please write this paragraph. Please write. It is important. Please write. When you write, please tell me. I continue. Okay, do you write? I continue or no? Okay. When everything for bacteria living make difficult in environment, such radiation, such cold, such hot, such heat. Bacteria, yes, they're important. Bacteria makes itself resistance. Okay, how? How make itself resistant? By endoscope. You know what I say? By endoscope. And what is endoscope? Endospore is a part of bacteria, a small part of bacteria that, like one coat bacteria, make one coat around of themselves to be resistant. To be resistant. Okay? You understand? I don't think it was difficult. It was very easy for you. Okay. You understand? Okay. Thanks God. Fatima, thanks God. Okay. And girls, how this endospore? I think it's go to sleeping. To sleeping. How it makes itself weight or girls. First, intends a basic naturals into the cell. Okay. Then conversion of these compounds into energy and vital cell constituents. Replication of chromosome, increase in size and mass of cell, division the cell into two daughter cells, each containing a copy of genome or overall components. Okay. It is important. It is important. And girl cell. We don't say about this. Continue culture. No. No. Oxygen. No. Metabolism. No. Okay. Okay. From this book, only study what I say today and last section. Okay. Do you understand? Only this. And other things not study, not in your exam. I told you. I take exam from slides, my slides, which I will listening or teaching you. Okay. Do you have any question? Do you have any question? Okay. Okay. Our lecture is finished. Thanks a lot of you. Don't worry about your exam. I will help you. And thanks a lot. Let's have another class. Our class means micro, I think. Okay.